Welcome back, everybody. This is Unit 4, Day 4, all right? So today we're going to be talking about diffusion. Remember, diffusion is a type of passive transport. So molecules are moving from high to low concentrations. And this process does not require ATP, does not require energy. All right, so today we're going to work through some practice problems about this. First, your do now says that molecules A and B are both organic molecules found in many cells. When tested, it is found that molecule A cannot pass through a cell membrane, but molecule B easily passes through. State one way the two molecules could differ that would account for the difference in the ability to pass through the cell membrane. All right, let's go ahead and pause this video, answer that question, and press play when you are ready to go over it. All right, so in this example, we see that molecule A cannot pass through that cell membrane, but molecule B can. This kind of sounds like something we did in lab on Friday, right? Molecule A couldn't pass through the cell membrane, so maybe, just maybe, that molecule is starch. And molecule B could easily pass through, so maybe that molecule is glucose, right? Both of these are examples of sugars, but they differ in terms of size, right? So something that would account for the difference in the ability to pass through the cell membrane would be size. So in this example, molecule A is too big to pass through. But molecule B is small enough to diffuse. All right, so the answer there is they differ in terms of size. All right, moving on to part A. You're going to use this picture that's given to answer questions 1 through 4. Okay, so in this picture, we see a sugar molecule or a sugar cube gradually dissolving, and those molecules are spreading out throughout the entire solution in this beaker. So the first question says, what process accounts for the change shown in lab setup A? So again, we see that this sugar cube is dissolving, and those molecules are being dispersed throughout the entire solution. So the process that accounts for that change is none other than diffusion. We see that the molecules are diffusing. They're going from an area of high concentration, and eventually they're spreading out throughout the entire solutions. They're moving to those areas of low concentration. Right, so sugar molecules are moving. From high to low concentration. All right, number 2, question 2 says, in lab setup B, structure Z prevents the movement of sugar molecules into side 1. Which part of a living cell serves the same purpose as structure Z? So here in lab setup B, we see that structure Z has been placed down the middle of this beaker. And we see that side 2, all of these sugar molecules are spread throughout the solution. But on side B, we don't have any sugar molecules. We only have water. And that tells us that these sugar molecules are unable to diffuse through the membrane. So what is structure Z? Which part of a living cell serves the same purpose as structure Z? Hopefully you got that that is the cell membrane, right? It's still a semi-permeable membrane, meaning that some things are able to diffuse through, but other things can't. And in this example, these sugar molecules cannot diffuse through. All right, question 3 says, in lab setup B, the sugar molecule that is not able to diffuse through structure Z can likely be identified as which type of sugar molecule? Support your answer. So because this is a sugar molecule, we're going to refer to it as a saccharide. And I seem to remember a vocabulary word that means a really big sugar molecule. Maybe it starts with a P. That is a polysaccharide, all right? So this sugar in this example is a polysaccharide, meaning it is too big The sugar molecule is too big to diffuse through the membrane. Okay. And finally, question number 4 says, explain what could be done to this sugar molecule that would allow for diffusion to occur. So we're dealing with a polysaccharide. It's a long chain of sugar molecules. It's too big to diffuse through that membrane. So if you think back to unit 3, what's something that had to happen to big molecules in order for them to pass through the membrane? Right? Question number 4, the answer is it has to be digested or broken down. Right? So the polysaccharide must be digested or broken down into smaller sugar molecules known as monosaccharides in order to be small enough to diffuse. Right, kind of had to squeeze that in there, but polysaccharides are too big to diffuse through the membrane. So in order for this sugar right here to diffuse, we would have to break it down into something smaller so it would be small enough to cross that semi-permeable membrane. Right. Next up, we see a diagram that shows us the before and after picture of molecules moving in and out of a cell. Now, whenever you see a picture that looks like this, I want you to stop and create a chart like the one I'm about to show you. But in this case, since we're looking at a before and after picture, we're actually going to create 2 charts. Right, so I'm going to do one side with before and one side with after. And the chart that I want you to make is going to look something like this. And I want you to label one side of the chart in and one side of the chart out. This side is going to be where we label the molecules that are found inside the cell. And this side is where we're going to label the molecules that are found outside the cell. And we're going to do that for both our before and after pictures. Right, so in our before picture right here, we're given a key to help designate what each of these shapes represent. And we're going to go through each of these shapes. So first we have our water molecules. I'm going to write those in blue. We're going to count the total number of water molecules inside and outside of the cell. So inside I see we have, oh no, it exited out. This always happens. There we go. All right, inside the cell, we have 1, 2, 3, 4, 5, 6, 7, 8 water molecules. Okay, so I've drawn 8 H2O molecules. And outside our cell, we have 1, 2, and 3. So I'm going to draw 3 H2O on the outside of the cell. Okay, for oxygen, I'll do that in red. We see that this is represented by a square. So I'll count the number of squares inside the cell, and we have 2. So we have 2 oxygen molecules inside the cell. And outside we have 1, 2, 3, 4, 5, 6, 7. We have 7 molecules outside the cell. And finally, protein, I will draw that in green. We have 1, 2 molecules inside the cell and 0 outside the cell. Okay, so now we have labeled the contents inside our cell from our before diagram. But we're also given an after picture. So we're told that over time, that is ugly. Over time, I don't know if that's any better, but over time we end up with this picture that we see over here. So we're also going to make a second chart that labels the contents of that cell. All right, so inside our cell, let's switch back to blue for our water molecules. We have 1, 2, 3, 4, 5, 6 molecules of water inside the cell. And 1, 2, 3, 4, 5 molecules of water outside the cell. All right. For oxygen, we have 1, 2, 3, 4 molecules of oxygen inside the cell. and 1, 2, 3, 4, 5 molecules of oxygen outside the cell. Finally, for protein, I still see that we have 2 molecules of protein inside the cell and 0 molecules of protein outside the cell. So what does all of this mean? Well, I want you guys to look at the number of molecules that were found inside and outside the cell for water, oxygen, and protein in both our before and after pictures. So we see that before we had 8 molecules of oxygen in, or I'm sorry, 8 molecules of water in and 3 molecules out. But after a period of time, we ended up with 6 molecules in and 5 molecules out. That tells us that these water molecules diffuse out of the cell in order to achieve equilibrium. All right, same thing for oxygen. We started with 2 molecules in and 7 out, but we ended with 4 in and 5 out. So that tells us that oxygen molecules diffused into the cell in order to achieve equilibrium, where we have an even number of molecules on both sides. But here with protein, we see that we started and we ended with 2 protein molecules inside the cell and 0 outside. Normally, molecules want to achieve equilibrium. They want to diffuse across that membrane so there's an even amount on both sides, but protein didn't. Why do you think that that happened? Well, protein was too big to diffuse across that membrane. So protein could not diffuse out of the cell because it was too big. Okay, so now I'm going to write that down. I'm actually going to pause this video so you don't have to sit here and watch like a minute of me writing, but I will press play as soon as I'm done writing. Let's see, how do I do that? All right, so you can see that I've written that water was small enough to diffuse out of the cell to reach equilibrium, right? We see right here that molecules of water were able to move out of the cell to maintain an even balance of both of water on both the inside and outside of the cell. Okay, I've also written that oxygen was small enough to diffuse into the cell to reach equilibrium because again, we see that we ended with an even amount on both sides of the membrane. But I've also written that protein was too big to diffuse out of the cell and could not reach equilibrium because we started and we ended with the same amount of protein inside the cell. It could not leave to achieve that equilibrium or that balance of molecules. All right, let's do this one more time. We've got one more practice question. Again, I'm going to make us a chart where we're able to organize what is inside and outside of the cell. So in this diagram, we're shown the relative concentration of molecules inside and out of the cell, and we're asked which statement best describes the general direction of diffusion across the membrane of this cell. Because this is diffusion, we are going from high to low concentrations. Okay, so let's label each of these molecules. So protein is a diamond shape. We have 1 molecule of protein inside the cell and 3 molecules of protein out. Right, for oxygen, it's a square. We have 1, 2, 3, 4 molecules of oxygen inside the cell and 2 molecules of oxygen out. All right, for glucose, it's a star. We have 1, 2, 3, 4 molecules of glucose inside the cell and 1 molecule of glucose out. And finally, for carbon dioxide, we have 1, 2, 3, 4, 5 molecules of CO2 in and 1 molecule of CO2 out. So now we know that molecules are going to want to travel from high to low concentration. So which statement best describes the general direction? Is it 1, glucose would diffuse into the cell? So let's look at our glucose. And we see that we have 4 molecules of glucose in and 1 out. So those molecules are going to want to travel out of the cell. So because this one says into, it's not A. B says protein would diffuse out of the cell. So we see that we have 3 molecules of protein out, 1 molecule of protein in. So those molecules are going to want to travel into the cell. So it's not B either. C says carbon dioxide would diffuse out of the cell. So let's look at our carbon dioxide. We see that we have 5 molecules in, 1 molecule out. Those molecules are going to want to travel out of the cell. So this looks like it would be a good answer, but let's check out D as well. And D says oxygen would diffuse into the cell. So we have 4 molecules of oxygen in and 2 molecules out. Those molecules are going to want to travel out of the cell. So it is not D. So our answer here for number 7 is C, carbon dioxide would diffuse out of the cell. All right, guys, that is all I have for you today. So go ahead and complete that exit ticket and make sure you turn it in before 8:30 tomorrow morning. Have a wonderful day, and I will see you guys soon.